So the final to look at is the response of vascular injury in terms of clotting and vascular repair. So the whole process of clot formation is taking a soluble protein and turning it into an insoluble protein, and then having that insoluble protein form cross hatches with itself so as to form what we refer to as a clot. The clot is regulated by a whole host of factors. You have the factors within the packet. Of the factors, the most important of these factors is excessive calcium within the plasma. Collection of calcium will trigger platelet activation regardless of actually having any type of vascular injury. Anytime we have stagnant flow, ions such as calcium can come out of solution, and this can trigger the clotting process. So let's take a look at the clotting process that takes place. So, intrinsic pathway is much more complex than the extrinsic pathway. The intrinsic pathway is predominantly regulated by calcium in the activation sequence, as well as two other activators. What ends up happening is that factor 12 gets activated. Factor 12 activation will then activate factor 11. Factor 11 activation will activate factor 9. Factor 9 will then activate factor 10. Factor 10 will take an inoperable hormone, prothrombin, and turn it into an operable hormone and enzyme, thrombin. These will then activate secondary pathways in a positive feedback loop, activating factors 5 and factor 7. Activation of factor 5 and factor 7 stimulates additional thrombin formation. Thrombin formation will then take a soluble protein, fibrinogen, and turn it into an insoluble protein, fibrin. The fibrin monomers will then start to cross-link with themselves, forming what's referred to as the polymer formation. Thrombin will then also activate factor 12. Activate factor 12 is what's actually going to trigger the polymer cross-link formation, which is what's going to trigger the clot. The entirety of the intrinsic pathway is regulated by stagnation of flow or inflammatory biomarkers coming from the squamous epithelium of the luminal tissue, triggering platelet activation. The extrinsic pathway is much simpler. It's so simple that all we need is some sort of vascular injury. Vascular injury activates 7 and what's referred to as tissue factors. 7 and tissue factors will then activate 10 and 11, and we have the same exact cascade of events that we saw within the intrinsic pathway. The reason why the extrinsic pathway is much simpler is that it's attempting to ensure vascular integrity. That way we will not have excessive fluid loss due to some sort of vascular injury. So before we take a look at these responses in terms of an animation, let's review a couple of key factors that are going to differentiate between the two pathways. The extrinsic pathway has fewer steps than the intrinsic pathway. It's a very rapid response. It's going to activate based off of tissue factors, as well as leaking of excitatory proteins. The intrinsic pathway is much more complex. It's very slow as it relates to the extrinsic pathway. There are several activators along the pathway, with the most predominant of those activators being excessive calcium in the plasma, stagnating, allowing for activation. It can activate with outside tissue damage. However, outside tissue damage is not necessary. Regardless of whether we're looking at the intrinsic or the extrinsic pathway, it's all about causing prothrombin to convert to thrombin by activation of the enzyme prothrombinase.
Even under physiological conditions, a small tear can appear in the wall of a blood vessel. In order to prevent blood loss, platelets and coagulation factors such as the pivotal factor 10, acting in a coordinated manner, close this wound. The platelets are responsible for a first sealing of the tear. Then, a number of coagulation factors are activated, leading to the formation of fibrin strands which stabilize the growing clot. These processes can also be triggered inappropriately and play a major role in the pathology of ACS, VTE, and AF. Thromboembolic disorders can affect both types of blood vessels. Arterial or white clots are primarily triggered by the rupture of an atherosclerotic plaque. Arterial clots are platelet-rich. Venous or red clots, on the other hand, mainly consist of red blood cells and fibrin. Vascular injury, hypercoagulability of the blood, and venous stasis play a crucial role in their development. It is important to note that the formation of both types of clots, arterial and venous, always involves platelets as well as coagulation factors. At the site of vessel injury, the first platelets arrive to start sealing the wound. Simultaneously, the coagulation cascade with its various coagulation factors is activated. This involves two pathways, the extrinsic and the intrinsic pathway. Extrinsic activation begins with now exposed molecules of the vessel wall, such as tissue factor, which forms a complex with factor 7. Finally leading to the activation of factor 10. This factor 10A is the point at which the extrinsic and the intrinsic pathways of the coagulation cascades meet. The intrinsic pathway consists of various coagulation factors activating each other in a chain reaction. At its end, a complex with an additional cofactor is formed. This complex now activates factor 10. Since the two pathways merge at the level of factor 10A, this factor has a pivotal role in the coagulation cascade. Further down the cascade, factor 10A in combination with 5A activates thrombin and induces the so-called thrombin burst. One molecule of factor 10A can catalyze the formation of 1,000 molecules of thrombin. These large amounts of thrombin cause the further activation of platelets and the enhanced formation of fibrin. Fibrin then forms strands, making up the mesh that stabilizes the platelet plug in an arterial clot and holds together the red blood cells in a venous clot. It can be concluded that the central role of factor 10A in the coagulation cascade makes it a viable target for therapeutic intervention in pathologically altered blood coagulation.
So let's take a look at the platelet activation sequence now. So with platelet activation sequence, what we're going to see is some sort of vascular injury here. This vascular injury is going to induce vascular spasming, which triggers the release of C-reactive protein, as well as platelet activating factors TF. This is also going to stimulate factors 5, 10, and 11, which is going to trigger the extrinsic response. So that's extrinsic response. Let's take a look at intrinsic response. So with intrinsic response, we have inflammatory biomarkers coming from the luminal epithelial tissue. This triggers activation of the platelets, which triggers stimulation of factors 9, 10, 11, and 12. Factors 9, 10, 11, and 12 start to aggregate platelets in the area. The aggregation of platelets triggers the spasming and the release of serotonin and prostaglandin E, which is going to induce additional clotting, leading to the full formation of the clot. Let's go ahead and take a look at this in terms of an animation now.
In a first step towards clot formation, platelets are recruited to the site of vessel injury by now exposed molecules of the vessel wall, such as collagen and von Willebrand factor. This factor mediates the linking of platelets to collagen via a specific receptor in the platelet membrane. The resulting change of shape of the platelet from its resting state into the dendritic form indicates activation. The activated platelet in turn releases prothrombotic molecules such as adenosine diphosphate, ADP. By binding to its receptors, ADP induces aggregation. And recruits further platelets to the site. Thromboxane is another important mediator of platelet activation and aggregation. Under its influence, the platelets cross-link with each other. These interlocking mechanisms cause platelet activation to snowball. The clot grows rapidly. Activated platelets also trigger the coagulation cascade and thus the formation of thrombin. Thrombin, in turn, stimulates platelet activation even further, a continuous feedback loop. Additionally, thrombin induces the formation of fibrin for the mesh stabilizing the clot. The self-reinforcing process of platelet activation, crucial in the formation of blood clots, is an obvious therapeutic target in conditions caused by inappropriately triggered blood coagulation.